So as we go through one by one through these structures, that will cover this objective. And so throughout the PowerPoint script, it starts with the mouth and then it ends up actually in part C, because remember chapter 23 has the parts A, B, and C for the PowerPoint scripts. And so throughout the entire presentation of this chapter, we go through each of these with their functions. Now remember, the digestive tract itself does not include the accessory organs, which are actually addressed in objective number 2. There are 5 what we consider accessory structures that actually add materials into the digestive tract. This does not include the stomach, and there are cells in the stomach, obviously, that secrete products into the lumen of the digestive tract. And if you'll remember, the lumen is the space inside where the materials travel. So the 5 accessory structures in the digestive system, we will start out with the salivary glands. Basically, this IE means that this is actually the answer as far as which structures I'm referring to. The salivary glands, they add saliva, and there's a discussion about saliva at the very beginning of the part A in the PowerPoint script for chapter 23. Then we get farther down. Of course, the stomach will add things, but we come to the gallbladder, the liver, the brush border cells, or I sometimes refer to as the cells in the lining of the small intestine, specifically the duodenum. These cells will be adding materials into the lumen of the duodenum, and finally the pancreas. So we have 5. 1 would be salivary glands, and then we have 4 that actually add materials into the duodenum. The gallbladder, the liver, the brush border cells within the mucosa of the duodenum, and the pancreas. So that is, those are the accessory structures that this objective is referring to. Then we get to the tissue layers. So throughout the digestive tract, there are distinct layers. Starting from the inside, from the lumen, the space inside, we have the mucosa. Okay, and the mucosa has a number of functions, specific functions depending on the area, but primarily the mucosa is a protective layer. There's mucus cells, goblet cells actually scattered throughout that secrete mucus on the surface of this membrane to protect the tissues from abrasion as materials travel through the lumen. And of course, throughout the digestive tract, the cells in the mucosa are going to vary somewhat. From the mucosa, deep to that is the submucosa, which provides an attachment or support for the cells of the mucosa. And then we have the muscularis externa, which is the main layer of muscle that is surrounding the organs of the digestive tract. And of course, this is what allows peristalsis and segmentation. And then finally, we have the serosa, which is the outermost layer of the tube that forms the digestive tract. And it's a moist, glistening appearance, and that actually prevents friction as organs are kind of moved around within the abdominal cavity. And that's all discussed right here. Chapter 23, Part A, slides number 14 and 15. Number 4, these are the major polymers or molecules of life, sometimes referred to as biological molecules, that are discussed generally in a fundamentals of biology course. That's a prerequisite for taking anatomy and physiology. I do review these at the very beginning of A&P 1 or Bio 263. Remember, they are the carbohydrates, proteins, lipids, and nucleic acids 3 out of 4 of those are actually food groups. So when we say polymers are very big, and they're typically made of smaller molecules that are actually joined together, and these are actually called, let me move this down a little bit, monomers. So the polymer, that means many, poly means many, and these are built with monomers. So the monomers that join together will create various polymers. With the carbohydrates, complex carbohydrates such as starches, cellulose, chitin, etc. Glycogen. Glycogen is actually a complex carbohydrate. And that's how we store a short-term energy in the liver and also in skeletal muscle. The monomer for carbohydrates, remember, is the simple sugar. We often call that a monosaccharide, the simple sugars such as glucose, fructose, etc. And then we have proteins, which are big molecules. They are considered polymers. Protein consists of many times it'll be more than one polypeptide chain, which is a chain of amino acids that are joined together through peptide bonds. So the polymer of a protein consists of a monomer, which would be the amino acid. Sometimes I refer to the monomers as the building block units. So if you kind of think of a train is actually many subunits. We call them train cars. So anyway, then we have fats or lipids, and they're big polymers. They're large molecules that are formed with a monomer called a fatty acid, and typically fatty acids are joined to another portion of a molecule. The example with triglycerides, which are neutral fats, often the fats we find in our diet, and also adipose tissue. These are called triglycerides. So there's the 3 fatty acids joined to a molecule of glycerol. So the building block for fats would be the fatty acids and glycerol, or different molecule. Keep in mind there's other fats, such as the cell membrane is made of phospholipids, etc. Then we have, finally, the 4th major polymer would be nucleic acids, such as deoxyribonucleic acid that makes up our DNA, our chromosomes, and then RNA, ribonucleic acid. Also ATP, adenosine triphosphate, is in that family. But the building block unit for nucleic acids would be the nucleotides such as adenine, thymine, cytosine, and guanine for DNA. It's slightly different with RNA, but you will find all of this in a review in our textbook. I believe it's chapter 3, but I also put up a PowerPoint that reviews these major polymers and the monomers that they are built with. Now keep in mind that these big polymers cannot get through the mucosa. They can't get through the cells and into the bloodstream. So they need to be broken down through what we call digestion. And there are many, many enzymes that are specific for each of the polymers and even parts of the polymers, such as, like with proteins, a protein gets broken down. Initially, the breakdown process starts in the stomach with pepsin, and then the polypeptide fragments will travel into the duodenum, and there's several proteases, enzymes, that will completely break down the polypeptides, remember the strands of amino acids, into individual amino acids so that they can be absorbed. So with carbohydrates actually starches, a particular starch called amylose, it's going to be broken down starting with saliva in the mouth. There's amylase. Then it will travel down to the stomach, and actually in the stomach you have the pepsin, which is actually secreted as pepsinogen, and then it's converted to pepsin by hydrochloric acid. And so you've got then finally in the duodenum. So when we say where are they broken down, where are they absorbed, the main place for even breaking them down is going to be in the duodenum. As soon as the chyme comes out from the stomach, it'll enter that short segment of the small intestines called the duodenum. Many enzymes are added by the brush border cells in the wall of the duodenum and the pancreas. Along with that, we see bile that's added. That bile is produced by the liver and then secreted and actually stored in the gallbladder. So that's also added to the duodenum to emulsify the fat so that enzymes called lipases can actually reach the molecules of the lipids and break them down. So that's where they are broken down, but they're absorbed primarily into the bloodstream. Now, the exception to that is that fats are absorbed into the lacteals of the lymphatic system. So, where they are absorbed and what they enter, it's primarily in the small intestine that they're going to be absorbed. And it's actually not only the absorption occurs all along in the jejunum too. So depending on how large of a meal someone ate, it will, as it travels through from the stomach into the duodenum and then through the jejunum, absorption is going to occur all along, of the monomers, primarily into the bloodstream, but of course the fats will be absorbed into lacteals. So that's this one. Number 6, why is it necessary for polymers to be broken down into monomers? Well, polymers are too large to get through the mucosa and into the bloodstream. So we need to have monomers because they're tiny and they can get through. Okay, number 7, describe and understand the various functions of the liver, including bioproduction and metabolic functions. There is a summary of liver functions, Part B, slides 61 and 65. This is not an exhaustive list of what the liver does. As you may have observed, the liver is large. It's huge. It has lots of functions. The liver cells are called hepatocytes. That means liver cells. And they have a number of jobs to do. So the liver will produce bile and secretes it. And the liver also stores liver glycogen, but there's a variety of metabolic functions. Remember, all the absorbed materials from the digestive tract will be sent right over to the liver through the hepatoportal system, and then that's going to allow the liver cells to further metabolize absorbed monomers, such as amino acids. So amino acids are not in their final form. Now they can be delivered to cells in the body that will be using amino acids to build their proteins. But amino acids, as they're broken down, they can yield actually urea. And so what's going to happen is the amino group, which is a nitrogen-containing group. Remember, every amino acid has the amino group and the carboxyl group. Amino group on one end will join to the carboxyl group of the adjacent amino acid to make polypeptides. So this nitrogen-containing group can be toxic. It can turn into ammonia. So the liver needs to convert that into a form that can be excreted by the body. The liver also can transaminate, and it deaminates amino acids, meaning it takes off that amino group, and transaminates, meaning it can actually take the amino group from one amino acid and transfer it to another one. Now there's only about 8 amino acids out of the 20 that our body needs to make its own proteins that we call essential amino acids. And essential amino acids we have to get in our diet. The liver cannot convert any amino acid into another one of these essential amino acids. So The liver has a lot of functions. The other thing the liver does is it conjugates bilirubin. If you'll remember from the lecture on blood, the breakdown product of hemoglobin is bilirubin. So there is an enormous number of functions that the liver does. And metabolic functions means changing molecules in order to make them less toxic. The liver will detoxify alcohol and other drugs, including hormones that are circulating in the body once they've done their job. So the liver is an amazing organ. 8. Peristalsis is the smooth muscle movements of the digestive tract that move materials along through the tract. Segmentation occurs primarily in the intestine, in the small intestine, where it actually will mix the chyme that has come into the small intestine from the stomach with the enzymes in which the fluid's there. So segmentation is a mixing effect. Excuse me. Number 9, describe what happens to a meal from consumption through the gastrointestinal tract or the digestive tract, and finally, release of the residue via the anus. This is just an overview of all of the things that are talked about in chapter 23. So that is an overview, basically going over the same thing. Number 10, list all the substances added to the digestive tract, where they're produced and released, the trigger for release, and their functions in the digestive process. That's pretty broad also. But it includes, here's somewhat of an answer or hints, it includes the mucus from the mucus cells that actually protect the lining from abrasion or damage. All of the products from the accessory organs that were described up in the, I think it was Objective 2, but also the stomach. The stomach, remember, has several cell types in there, and those gastric pits. There's the secretion of hydrochloric acid and pepsinogen, which is the inactive protein digesting enzyme. It needs to be converted into pepsin in order to start the breakdown of proteins. There's also, with the stomach, the mucous neck cells, which actually make a somewhat of an alkaline mucus, because remember, in the stomach environment, it's very acidic and it can damage the stomach lining. But if the mucus is somewhat alkaline, it will neutralize that acid. Okay, chemical digestion of proteins. This is actually, I'm just giving you also a little clue right here. Chemical digestion in carbohydrates, proteins, lipids, and nucleic acids are described in the PowerPoint, which I put up in your module, chapters 23 and 24, the summary of chemical digestion and nutrition. So this is the one that I went through very quickly in lecture, and it actually has tables in there that show you for each of the polymers, carbohydrates, proteins, lipids, and nucleic acids, how they're what enzymes are involved, what substances may be involved with breaking them down, and then where they are absorbed. Okay, number 11. This is figure 23.17, which I highlighted during the lecture. It's the big figure that has the cephalic, gastric, and intestinal phases illustrated. On the left-hand side would be factors that stimulate, and on the right, the factors that inhibit. So understand what's going on with that figure. And I explained that during lecture. So I'm not going to go over it right now. If you have any specific questions about that, please let me know. Just in summary, cephalic is from the brain. So this happens before food even hits your mouth. It's actually, it can include the other sensory organs, smell, thinking about food, and looking at a very beautiful presentation of food, etc. Gastric phase is when when anything hits the stomach, some kind of a nutrient. Intestinal phase starts in the duodenum, and that's when food begins begins to enter the duodenum. 12. The various cells in the stomach. This is in part B, I believe, of the PowerPoint script, and I kind of give you some hints here You have your mucous neck cells that secrete mucus, an alkaline mucus actually, and then you have hydrochloric acid that is secreted by the parietal cells, pepsinogen by the chief cells. This is all going into the lumen, or the space in the stomach. Gastrin is secreted by one of those enteroendocrine cells in the stomach, and this actually does not go into the lumen, but it goes into the bloodstream. So that's why it's called, the cells are called endocrine, because the product will go into the bloodstream. It doesn't travel throughout, it doesn't act on distant locations, but it acts right back on the stomach muscle. Gastrin actually, the stomach's gastrin increases stomach motility. And then in order to convert pepsinogen into pepsin, it requires the hydrochloric acid to be present. 13. Okay, intestinal gastrin, secretin, cholecystokinin, CCK, and vasoactive intestinal peptide, VIP, all of these initially, from the first burst of gastrin, you have an increase in stomach activity, but all of these intestinal endocrine products will go to the stomach and slow it down, because you can't have the entire stomach contents emptied into the duodenum at one time. There's little spurts of the chyme that will enter the duodenum, and that gives the duodenum time to digest the materials that have entered it. So the intestinal gastrin, secretin, and cholecystokinin, and VIP, all of these will actually regulate the stomach activities, basically decreasing stomach motility. These are also identified on this figure 23.717, and that also talks about their function. Cholecystokinin got its name because cholecysto refers to the gallbladder, and at the same time that it will act on the stomach to slow it down, but it also will stimulate the contraction of the gallbladder, because often it's the lipids that have entered the duodenum that will be the trigger for the release of cholecystokinin. And in order to emulsify the fats there, break the large droplets into smaller droplets, you need bile, and that would be stored in the gallbladder. Okay, post-absorptive and post-absorptive phases. These are actually, I included the figures lower down on the study guide. Absorptive, of course, just means while you're eating, while you're absorbing food. So it'd be when you start eating, shortly for the next couple of hours, when you're absorbing the food, it will actually keep your blood glucose level high so that the body cells have plenty of nutrients to be able to use them, including the other monomers such as amino acids, etc. It will stimulate the storage of energy as well. So if you're taking in more food energy than you need, you're going to start storing energy. It will replenish any glycogen stores in the liver or skeletal muscle, and then it will begin to build adipose tissue. Post-absorptive phase just means after, when you're not absorbing. So you can think of this as the fasting phase. So when you're not eating or absorbing nutrients, how does the body keep its blood glucose within that normal range? Why doesn't it drop down? If it drops too low, you can actually die. So the body has ways to keep that blood glucose at a normal level through the breakdown of glycogen. That's a short-term complex carbohydrate that the body has that can be broken down in order to release glucose into the bloodstream to keep the blood glucose within the normal range. After that, if somebody is fasting for a long time, they will begin to break down their long-term stores of energy, which would be the adipose tissue. If somebody is starving for a long time and not taking in enough food energy to meet their energy needs, they will actually begin to start breaking down their own body proteins, such as skeletal muscle. So that is what that refers to. And then the last objectives right here, whoops, this actually, number 15, 16, and 17, we're not quite to the end yet. This is referring to in the liver. Okay, so lipids are metabolized in the liver. Protein metabolism, this also occurs in the liver, and so does nucleic acid metabolism. Now, metabolism also includes digestion, but it takes it several steps further. For example, with proteins, amino acids, individual amino acids need to be deaminated by the liver, transaminated, etc. There's a lot of things that can happen with that. The amino group, the nitrogen-containing group, needs to be turned into a less toxic form because it will produce urea. And actually, the body excretes that in the urine. Hence, urine is named because the major waste product is urea, urea, which is a byproduct in the breakdown of that amino group of protein, of amino acids. Nucleic acids, they actually can form what we call uric acid. Remember, nucleic acids also have nitrogen. So we call these together nitrogenous waste, and the body needs to eliminate those. Okay, the functions of the large intestine, I basically went through those. This is to store feces until expulsion, and so water would be reabsorbed from the large intestine. Water reabsorption actually starts in the small intestine, but it continues from the large intestine. The major highlight of the large intestines is that they are filled with on the mucosa, in the lumen, with a whole variety of bacteria. We call these your microbiota, and you may hear about probiotics. These are the good bacteria, the bacteria that are actually friendly once if they stay there, as long as they don't get into your bloodstream or directly into your body tissues. They're helping us be healthier by allowing us to absorb certain vitamins, such as some of the B vitamins and vitamin K, which is necessary for blood clotting. So they also take up the space, the lining, to prevent establishment of some kind of a pathogen that may enter into your body through the gastrointestinal tract. So now I added these two figures because they're just so pretty. Figure 19B. Anyway, this is just showing you absorptive state. So this is the time where you're absorbing glucose, amino acids, and fats. So what happens to them? Well, this is going to keep your blood glucose high, and it will actually, hopefully it's not going to be too high. What happens when somebody has diabetes mellitus is their blood glucose goes abnormally high because the glucose cannot get into the body cells. It stays in the bloodstream. So when the blood glucose goes up, this glucose enters the body tissues and allows cellular respiration. In this case, it's aerobic cellular respiration, so glucose plus oxygen will be broken. The glucose is broken down by body cells in order to get ATP energy, and then carbon dioxide and water are released. If you have more energy than you need, the body will begin to build adipose tissue. And so, and this is pretty much an ad infinitum process. There's no real homeostatic balance. So any kind of extra energy, if you're taking in more food energy that you're using, it will begin to build body fats. Now, before this happens, actually, if your glycogen stores in the liver and in muscle have been depleted, the glucose will first be bound together to make glycogen stores, because this is your short-term energy storage in skeletal muscle and also in the liver. So you can see glycogen is made here. So basically that's what I want you to know about for the absorptive stage. And then the blue background one down here is post-absorptive. And keep in mind, this is also known as the fasting state. when you get up in the morning, you have break fast or breakfast because you are not eating all night while you're sleeping, theoretically. So right here you can see, the blood glucose will be dropping, and so the body, when blood glucose drops, it sends a signal actually to the pancreas to release glucagon. Think of the glucose is gone, and it's going to target the liver to break down glycogen to release the glycogen, to store glycogen will be turned into glucose, and that will release the glucose into the bloodstream to keep the blood glucose up. Also, long term, the body will begin to break down adipose tissue. So this would be when you're not eating for a day or two and you're still expending energy. So your glycogen stores have been exhausted, and now the body will move toward the adipose tissue and start breaking it down. And what that does is it yields fatty acids and glycerol. These are the building blocks. I don't know why that's shaking. But anyway, these can actually be used for energy by the brain. And also, but you do get, with the breaking down fats, you get the production of keto acids, which need to be metabolized by the liver. And so you have keto acids also here from amino acid breakdown. So just keep in mind, during the fasting stage, the body will then convert to breaking down its glycogen stores and then adipose tissue. So, and that is pretty much it. Have a wonderful day. I will see you soon. Bye-bye.